Are we on? Da-da. Current. I never know if we are on or not. That's interesting. We got our research back. I think we're on. Okie dokie, let's start rolling. I'm actually very excited about this, like classes, virtual classes. I know they super unstructured. That's maybe a blessing and a curse. I'm pretty sure that if they were a little bit more structured, we would progress way faster, or maybe not. We have a lot of tabs open. Yeah. And we started on this question number 6. We will continue this question. I like it so far, maybe for like a week or so. Then we will define our profile. I mean, our mine, and maybe we will adapt. How should I learn that faster? So far, I'm so pumped. And yes, yesterday we started a research, and I can publish this. So the convergence of AI and cellular biology has entered a critical inflection point. I love it. That's what we're doing. Cellular biology meets software, a field guide for 2026. I love this. I know it's like AI is usually over optimistic, and we need to do a reality check on this, but this is interesting. So foundation models now predicts proteins structure. Yes, that's a alpha fold, I believe, right? Alpha fold. That's it was big breakthrough how the protein folds. But I didn't know like is this is this is this how to make it full screen now? Hello. Publish, download it. Let's go publish. Research public. Publish to web. Anyone with a link can view, and your artifact may show up in search engine view. Chat will stay private. Maybe. For now, I just want a bigger one. Oh, here we go. Got it. Okay, we're gonna keep it here. We're gonna skim through this. Oh no, it's gonna take me like 3 days to read this. Whoa. Should we? Oh my god, that's gonna take me a week. 1 week to read this. 10 points. Okay. Let's try to skim it. Foundation models now predict structure, experiment, and accuracy, novel genomes, and generate novel genomes. Okay. Do I remember what is genome is? Genome. Vocabulary. let's see, telomere definition, repetitive DNA cap at end of linear chromosome. Chromosome. I don't even know what's chromosome. I mean, I heard that word many times, but if you ask me to draw it right now, like pen and paper, I wouldn't be able to draw it. I feel like that's something like this. One of these is chromosome, or maybe pairs, it's chromosome. Etymology, Greek telos and meros, part, domain, molecular biology. Like, I need to remember how I learn English. Oh. So when I open too complex a book and I don't understand like 50% of the words of the on a book in a page, it means it's too complex for me. I probably should use same strategy here. Search, refresh. Where is my genome? Genome. Complete set of DNA in an organism. Oh, right. All chromosomes, all genes, plus non-coding regions. The full instruction manual. So that's basically like, if we compare to AI, everything, weights, harnesses, system prompts, model, everything, right? Anyway, Messenger Cell shared latest, yet the field's most hyped application. Prediction how cell responds to perturbation. Explain me like I'm 20 years old. I should have already that prompt preset, but anyway. By the way, Opus 4.7. Think of cell like a tiny self-regulating city. It has its normal rhythm, making proteins, producing energy, mitochondria, dividing when needed. That's my question. Like, what's trigger for cell to decide, or when cell decide to divide, to split? A perturbation is anything that disrupts this rhythm, like heat, toxin, viruses, nutrition, starvation, mechanical stress, or even a drug you took. Sensing the problem, cells are covered in and filled with sensors called receptors. I know that. At least I know something. Some sit on the other outer membrane, watching the outside world. Yes. Is there food? Question mark. Is something attacking me? Question mark. imagine like the dog biting your leg and you're like, is something attacking me? Others are inside watching internal conditions. Is my DNA damaged? Am I running out of energy? Are my proteins misfolding? That's a good. These sensors are usually proteins that physically change shape when they detect something, and that shape change is, and the shape change is the alarm signal. Okay, signal transduction. By the way, very good explanation. Perfect. Okay, that's too complicated for me. For now, the response itself, the signal. By the way, we know what's cell perturbation means.
Perturbations. Perturbations.
Perturbations. Sounds like in a plane when you go through the rough air. Still falls to be a linear baseline. These tensions between extraordinary tool building success and stubborn biological complexity. Why do you call it stubborn? It's just, I think it's pretty. Like complexity that biology created, or organic complexity, it fascinates me. Defines the landscape of April 2026. For a Go developer on a Google Cloud looking to enter the space, oh yeah, that's me. That's me. The opportunity is real, but demands clear-eyed understanding of what works. You see this eye? It's very clear. What hype and where the hard problems remain. Yes, lay it out. Lay it out for me. The stakes are enormous. 3.8 billion followers of the AI drug discovery in 2024, along with the single rounds exceeding $1 billion. DeepMind's AlphaFold, yeah, mentioned it, called it on the Nobel Prize. Oh, really? Yet no AI discovered drug has received FDA approval, and multiply independent evaluations show that deep learning models for cellular response prediction perform no better than averaging the training data. Oh, so you train a model on something, and it couldn't predict anything better. It just average of what has been trained on. That's a ceiling right here. I love this. That's a field of opportunity. This reports, it means you need novel approach. Like what this sentence mean to me? Performs no better than averaging the training data. This sentence call for novel approach. This report maps the entire terrain, models, technique, companies, datasets, gaps, and the annotated reading list of landmark papers. Okay, I know what we're gonna do. We're gonna take this research, obviously it's insane, and we're gonna create a Karpati obsidian graph from it. We have to do it. For now, we have to start from that, and then we will evolve it into something maybe even cooler. See, I don't know what to do. Continue reading this or go back to the questions. The most ambitious frame of field company demands that the cell perspective. 42 researchers from CZI, Stanford, EPU, Tech, Google Research, Harvard, and ARC. So many smart people. Imagine might meet some of them or all of them. Imagine like take a hand of each of these researcher. I would be star. Would be so excited. Need to find the conference where they go. The concept of AI virtual cell, AI VC. Oh, interesting. I like this. I like that concept. I want to build one myself. Envisions a multi-scale, multimodal neural network that can represent and simulate behavior of molecules or cells and tissue across diverse states. That's deep. So they don't simulate just organelles and behavior of the cell itself. They went even deeper. This reminds me What does it remind me? So respond to perturbation. Solaris. Yeah. Stanislav Lem. That's a amazing book, by the way. Is it a movie? Maybe. But what explains there is that the terrestrial species, like basically other life, recreated our life as simply by mimicking our molecular structure. But it can do even smaller, way smaller. That's it. I explained the whole book in one sentence. Yeah, we're gonna do this later. Let's go with the question. I need to know how bad am I? How bad am I at what's my baseline, right? So, and then what my type, so I can catch up, because I need to catch up with those 42. Like this 42, I need to catch up with them. Codons. I remember what codons are. Let's say in the neighborhood. Let's stay in the neighborhood. Since you are warm, what's the difference between DNA and RNA? DNA is the whole instruction set for all the proteins that can be made, all the amino acids, maybe. Basically, that's a whole set of instructions. 3 millions unique combinations in particular order. RNA, however, it's a small chunk of it, but not just a chunk, it's a transportable chunk. So RNA created from a subset of DNA, so it can be moved to different area to then create a protein. RNA transport information from DNA, and then from RNA to protein, I believe that's what's translated. There's two things, transport and translate. I don't remember which one is information flow. Yeah. And I remember this arrows was, it's a dogma. What is that? Yeah. Transcription, sorry, not transport. Transcription from DNA to RNA. DNA is a storage. RNA is a messenger. So it's transcription. And then from RNA to protein, it's translation. But that's we should not stay in the neighborhood for the next time, because I saw the answers like just scrolling up. Don't do that. We're still on the Opus 4.6. Need to switch it to 4.7. That's fake because I just read it. DNA set storage, correct DNA, portable copy of subset correct, RNA transcription correct. What you mean? The actual molecular differences. DNA is double stranded, the famous double helix. RNA is usually single stranded. Oh, right. DNA uses a sugar deoxyribose. RNA uses ribose. Ribose? I need to catch up with the vocabulary. What's that?
Ribose.
Ribose. Yeah, my bad. Ribose. Deoxyribose, RNA use ribose. One extra oxygen atom. That's the deoxy part. Deoxy has one extra oxygen, and ribose pretty much the same. DNA use bases A, T, G, C. RNA swaps out T for U. Uracil. Wow. We're going deeper.
Uracil. Uracil.
So when we convert from the storage to messenger, we have to swap out one element, but only one. That's so strange. So RNA pairs are AU instead of AT. DNA is stable and stays in the nucleus. RNA is temporarily made, used, then destroyed. By whom? Who destroy RNA? My questions. What are the all possible combinations of and Yeah, I need to remove that. So length 1 for ATGC. No comments. How is the length? Each position can independently be any of 4 bases. That sounds like we have 2-bit in computing and we have 4-bit in organic matter. That's way too close to be random coincidence. ATGC, A8AT. So the T switch to U, AG, AC, TA. Can it be UA in RNA? These are condos, the units that's code for amino acid. This grows exponentially fast. A human genome is about 3 billion bases long. So the number of theoretically possible human length sequences is 4 in a 3 billion in the power of 3 billion, a number with nearly 2 billion digits. Whoa. That's a scary number, but why are we looking to that? I think it's misleadingly scary, meaning like I don't know, but it's giving me a sensation that's like imagine a LLM, right? 1 billion parameters. So you have 1 billion parameter, and then each parameter can fluctuate from a 0 to 1, let's say on a scale of 10. 0.1, 0.2, 0. So there are 10 options for each parameter. That's what you have 10 in the power of 1 billion. But you never measure a model or LLM or how different it could be just by multiplication of all possible options of those parameters by the number of parameters. It's kind of useless. Like, why even giving this? Like, this is noise. If you just want to pick bases from ATGC without caring about order with repetition, therefore the formulas is Cn plus 3 multiplied by 3. Pick 2, 10 combination, pick 3, 20 combination, and pick 4, 35 combinations. This is rarely what people usually mean in biology. So because the sequence of order matters enormously. Yeah, you know that. The sequence and order is the most important. ATG is start codon. TGA is a stop codon. Oh, okay. So ATG and TGA is kind of entry and exit point for each codon. Same letters, completely different meaning. Just the pairing rules. That's what I was looking for. Okay. Double-stranded DNA A always pairs with T. Okay. In double-stranded DNA, A always pairs with T. That's exactly what I was like. Yeah. This just got me on a sidetrack. Unneeded sidetrack. A always pairs with T, and G always pairs with C. GC, AT, GC. Okay. So there are really only two types of base pairs, AT and GC. This is why when you sequence one strand, you automatically know the other. So when you sequence one helix of double helix, you know automatically the other, unless you sequence RNA, because RNA has only one. What's the word? Strand. Okay. So RNA pairs are AU instead of AT. DNA is stable and stays in the nucleus. RNA is temporary mated. Yeah, now it's good. Now I got it. I learned something today. That RNA, it's one strain instead of two. It's temporary. I knew it's smaller. It made, then destroyed, but I didn't know about T and U. That's T replaced for U. What is a natural selection in your own worlds? Natural selection. Natural selection, it's a mechanism by which DNA and species, animal, plants, all of them, they have small mutation. Maybe on every generation, maybe on every other generation, maybe it's random, maybe sometimes mutation is bigger, some like more often, sometimes it's less often. I don't remember the factors, but there is always mutation. And also with a sexual reproduction, it's a mix of different DNAs, X and Y chromosomes, and that's give different variations on the end results. So by changing the variables, you are getting results a little bit different results. And then those results are actually play out their lives in the real environment. And the variations that more adapted to the environment survive, and variations that's useless or not matching the environment die out. Therefore, generation after generation, natural selection will pick only the one who fits the most for the current environment and current circumstances. And I believe that Darwin did it with the with the birds, where he noticed the peak was adapted for different flowers on the islands when the species was the same, but when they split it to the islands, they start adapting to each island individual plants and flowers, basically environments, and the peak starts changing generation after generation. How is this answered? Let's see. What's my score? This is near perfect answer, genuine. Yes. Nearly. Oh, still a lot of work. Mutations create variations, correct. Sexual reproduction mixes DNA, creating more variations, correct. Variations then feed the environment, survive, other die out, correct. This is the correct mechanism. Generation after generation, selection accumulates, correct. This is what makes the powerful, tiny advantages compound over deep time. Darwin's bird big islands, correct. Damn. Darwin's finches, yeah, on the Galapagos Islands. I wanted to say Galapagos, but I did. Different big shapes for different food sources. Seeds inside cactus, not just flowers, but close enough. Hey, this is the textbook example. I did my homework at school. Let's put it this way. I mean, just joking. I never did my homework. I hated homework. I don't know how anyone actually can do homework. Like, you study in a boring school. I mean, depends if it's boring or not. It really depends on the teacher. But you sit there for like 40 minutes, one class, and another 40 minutes, like 6 classes. It's almost 5 hours a day. You sit there, and then you go home, and you have to do homework. No, it's so painful, so insane, so not logical. I never did my homework. I developed a strategy early. I basically listened what the teacher was saying, and next day, not like week later, just next day until I remember everything, I just repeat word to word. And that's it, like A grade, good job, Vitaly, and I don't never need to do homework. Okay, minor refinements, not errors. Mutations happen every generation in every individual. No, that's not correct. Most are neutral, do nothing. Some are harmful, rarely. Once is beneficial. The ratio matter. Most variation is noise. Selection finds the signal. Mutations happen every generation, in every individual. That's I didn't know. Yeah, this I didn't know. Yeah, but interesting part, I'm pretty sure mutation happens also not by generation when you reproduce, but also on a division, on a cell division, mutation might happen. I wouldn't say this is the main cause of cancer, but I'm pretty sure that's what's happening. I need to double check that. X and Y chromosome specifically determine sex. The mixing you are describing is more broadly called recombination during meiosis. So much to learn. Vocabulary. Meiosis. The thing is like to learn on a different language. It's also a little bit tough, but at this point, I don't know which language will be easier for me to learn. Chromosomes from both parents shuffle and swap segments. XY is just one pair out of 23. Yes, the company 23 and me, right? You described adaptive radiation in the fish. No species split into many to fill different niches. That's a specific parent produced by natural selection, not natural selection. Oh, not natural selection itself. Yeah. But you clearly understand the mechanism that drive it. Okay. What? Ducking a tiny bit only because of XY chromosomes isn't quite the right framing for genetic mixing. True. Otherwise, textbook quality answer in your own words. Okay. I got 56 above chance. So basically, if you throw the coin, I'm a little bit better than a coin throw. Do I have a coin? Yeah. That's a coin, and that's me. New territory. What is the immune system's job, and how does it know what to attack? Okay, immune system job is actually identify, I would say not molecules, I would say proteins that not belong to your own body. So some kind of external protein that harmful for organism or should not be there. And it's basically identified and trying to surround it, I guess, and then take it away. I think that's how it's fighted. And how does it know what to attack? That's interesting. I think it's know what to attack by actually storing a little chunk or copy of that virus DNA on its own DNA. Maybe not DNA, but it's actually store a little bit of information about that virus or bad protein. Maybe the whole protein was wrong, so it's attack viruses, not the proteins. I need to find the difference. Yeah, that's exactly right. It doesn't attack the proteins. No, that's stupid. It attacks viruses. How does it know that it's virus? Because it stores a signature of each virus on its own, and I don't know where, on its own storage, I would call it, but its storage probably, I wouldn't say it's DNA, or maybe it's something similar to DNA. So it's basically, maybe it's a RNA. No, not RNA. It's stored somewhere, and that's what I'm thinking. That's why it's know how to attack, and that's why the shots works, right? So like that's protecting us from diseases. It gives us a small amount of viruses, so that our white cells can attack it, store a little bit, so next time it can recognize faster before it went viral. That's the whole point of vaccination. Really good reason here, especially the vaccination logic. Let me untangle what you got right from where you got tied yourself in a knot. By the way, I don't know how much should I trust these answers, because AI lied me a lot in the past. I guess until I run into production issue when I say something stupid in public, with a straight face and someone call me on it, then I will know that AI lied to me back in day 2 or day 1. I'll find that reference and I will feed that to a new model of AI so it can correct it for others. About pass surrounding and remove identifiers that doing below. Okay. Identifies things that don't belong. Correct. This is the core job. Surrounds and removes threats. Correct. White blood cells literally engulf invaders, called phago cell eating. Phagocytosis. I like this. What's the name when cell eat another cell? It's called phagocytosis.
Phagocytosis.
Phagocytosis. Phagocytosis. Store information about past invaders. Correct. This is called immunological memory, and it's the whole base of adaptive immunity. Vaccination equals small harmless version of the system can learn the signature. Correct, you nailed it, the logic perfectly. Where you got tangled, the protein things was actually right the first time. Oh, really? Interesting. The immune system doesn't recognize whole viruses directly. It recognizes proteins on the surface of invaders. Oh, these surface proteins are called antigens. So your first instinct identifies proteins that don't belong was more accurate than your correction. No, it attacks viruses, not proteins. It attacks viruses by recognizing the surface proteins, antigens. So if my nanobot that I'm gonna put into bloodstream will not have any antigens, then it will be taken as bias, right? That's what's up. It attacks viruses by recognizing surface proteins, antigens. I need to know antigens. Antigens. Your own cells also have surface proteins. Antigens, images. Yeah, so that like famous COVID virus with all these like spikes. Those spikes are antigens, right? Antigen stacks, yeah. And this is the Y. What is Y stuff? And Y1 is the antibody. So it's when it fits, like, oh, I know that's a protein, that's an antigen, and I need to surround it, like engulf it, and not engulf it, but
Phagocytosis.
I need to do some phagocytosis on that cell that has a lot of antigens. Where's my antigens? Come on. Yeah, antigens. So I can do again
Phagocytosis.
phagocytosis. An antigen is a substance that's induced the formation of antibodies because it is. What do you mean substance? It's a protein, right? Antibody, antigen, pathogen, different antigen. All right, so pathogen can have multiple antigens. An antibody will store the shape of antigens. So the question now, what is pathogen? And I'm assuming pathogen is something bad. Antigen, bacteria, yeah. antibodies, antigen virus, B cells. Okay, it's way too deep. I'm sorry, I need to fix that coughing stuff. Okay, so your first instinct, your own cells also have surface protein. Your immune system learns earlier in life, these are mine, ignore them. So the whole situation with what's that really bad disease, anti-immune disease. So what happened is our own surface proteins, which are antigens, for some reason start to be recognized as not a good one. And our own immune system attacks our own parts of body, our own cells, just because it's thinking our antigens are not our, but it's external. And it's not ignoring them. Anything with unfamiliar surface proteins gets flagged. Yeah. Okay. Day 2. Do you know how anti-immune disease works? Yeah, it attacks its own body. Why? Because it simply forgot that protein that's sticking out of our cells, that's called antigens, are actually our. It thinks it's external and trying to exterminate it. So fix the memory of antibodies, fix the memory of immune system, that these antigens are our and this one not us. Disease is fixed. Probability of storage you were reaching for. After fighting an invader, your immune system creates memory cell. Yeah, tell me more about this, because I was lacking a type of white blood cell called memory B cell. Oh, and memory T cells. Yes, I remember that. Oh, I have T cells now, right? I have T cells for COVID. I have T cells for that. Okay, so I have a memory. So the question is, why don't you have T cells from a human A and put it into human B or animal, whatever? If it's same T cells, you don't need vaccines. You just transfer the memory. Like, for example, like, hey, Bob, you don't need to learn on your own through fighting this virus or whatever. You don't need to learn your own with your own immune system. Here's my memory, take it. I have basically a collection of all T cells. So now instead of vaccination, we can just transfer memories. And by the way, I'm learning, and all my hypotheses here are just random connections. That means nothing. So I'm sure what I'm saying is probably not possible. You cannot just transfer memory or T cells from one species to another, not species, but like even with the same relatives. I'm pretty sure like something bad happened. Actually, let's Google it. I want to know. Can you transfer memory T cells from me to my brother so he does not need to do immunization? Maybe it's just technically not possible, but theoretically it should work. But the answer no, yes, that's what I thought. This doesn't work in practice. Okay, it doesn't work in practice, but theoretically, we're just not practicing it well. The idea is scientifically reasonable on the surface, yeah. See, like all my ideas are on the surface, and it's okay. It's a day 2. Like, look, day 2. Memory T cells are exactly what vaccines train. and transferring them would theoretically transfer the immunity. Yes, but there are hard biological roadblocks that makes this non-starter for routine use between siblings. The HLA matching problem. Okay, so I would guess each T cell for each organism a little bit different. Therefore, the T cell would be recognized like invaders again and will be rejected. Your T cells recognize infected cells through a protein complex called HLA, human leukocyte antigen. Okay, every person has a nearly unique HLA signature inherited from their parents. Your T cells are trained to work with your HLA, not your brothers. But how about my parents then? If transfer two bad things happen, graft viruses host diseases. Whoa, GVHD. Your T cells see your brother's entire body as a foreign and attack it. This. Oh yes, that's what I thought. Yeah, it's thinking the T cell itself is different because the signature is different. So it's kind of what would become comparison like in software, right? Not the sum check. It's interesting. It's like SSH key? No. Salt and paper? No. Very interesting. Like, I need to think about it. It's fascinating. I will ask about the parents for sure, but rejection, your brother immune system see the T cells and kill them. So the transfer immunity disappear within days or week anyway. It's often fatal. The T cells see your brother entire body as foreign attack. This is that's so basically if we fix HLA, if you take memory with HLA-A and convert it into HLA-B, then it will work. Siblings have only about 25% chance of being a full HLA match. Yes, but the parents then, right? Then parents is 50. What are the percentage of HLA match between child and parent? By the way, when we do clones, HLA will work. So we don't need to immune every single clone. We can just transfer memory. Even with the match, minor antigen mismatches still cause problem. This is why bone marrow transplants require intensive HLA matching. Immunosuppression and still carry serious risk. Okay, this is I I didn't read the research of those 42 researchers, right? I will read it tonight. Actually, I'm lying. I have to work tonight, but I will read it on the weekends. But looks like this is a real problem that's asking to be solved. So right now they do transfer it sometimes, but they push a lot of immune suppression, kind of like, hey, antibodies, chill. Don't look for invaders, all right? So the bone marrow can actually work or like adapt or not be rejected. Layers. Hold on, I was not here, right? Okay, currency, that's yesterday's stuff. Where am I? Oh, here we go. Why vaccines are better anyway? Even if the transfer work, vaccines produce immunity that lives in the right tissues, lymphs, not mucous. Oh, I didn't know that immunity lives in different tissues. Includes both T cells and B cells producing antibodies. Gets okay, so that image after all was not bad. So it's a B cells. Okay. So B cells release these antibodies. Oh, now it start clicking. The B cell, it's actually a cell. And what it does, it's producing those antibodies inside of the cell and then pushing them through my brain to go swim and attack antigens, bacteria, antigens of bacteria or any viruses, not viruses or bacteria themselves, but antigens of those bacteria and viruses. And I guess the B cells would be excited, meaning will start produce more antibodies when something is detected. Interesting. This reminds me, I don't know why, but water economics represented as a water machine. Like, that's how my memory works. I don't know why, but it's economics presented as water. Yes. Philips machine. Yes. Philips machine. It's amazing machine. It's pretty cool. So different liquids represent different concentration or amount of stuff, and then they all balance out of each other. I think this guy is saying it's the best. 52 minutes, that's too much.
I'm at the University of Cambridge. That is the world's only working water-powered model of the British economy. And by moving this graph, we'll find out what happens when you divide by zero. For the sake of this week's Prime Minister, don't try this at home. Huge thanks to NordVPN for sponsoring this episode. Time before digital governments pay for projects, banks invest funds, and our flows
so he explained it here.
Some of this expenditure is water is pumped to the top as income. Some is taxed, going to the government. Some is saved to the banking sector, and the rest is spent. Governments pay for projects, banks invest funds, and our flows recombine. Some of this expenditure is on imports, and money goes out the economy. Some extra funds trickle in from exports, and everything meets at the bottom as transaction balances. The cycle begins anew.
That's it. Explained in 30 seconds. Go back. I'm getting lost in this, but score. Free type. Okay, where am I? Vaccine better anyway. Okay. So I was looking at this image and I was thinking like there is certain concentration of antibodies of particular B cell, let's say for coronavirus, right? In the body, swimming around. And as soon as the amount of them become less because they bounded to the bacteria or virus, these B cells become like, hey, looks like my antibodies doesn't exist in the bloodstream anymore. I need to produce more of them. And like, let's go. That's my assumptions. And understanding, I may be completely off. That will not explain how the immune system might forget stuff, because why you do second or third vaccination so you're not forgetting? Because if it's only one, the concentration eventually will go down to 0 and will not be produced by B cells anymore. The B cells might not exist anymore, and therefore you will not have immunity to that antigen anymore. And pathogen will attack you. Damn, I'm start using the cool words. I like that. 2 layers of DNS storage where we are. Okay, we're not here. My cell biology day 1. I don't even know what's this. OBS. Yeah, this one. Or why vaccines is better anyway? Even if the transfer work vaccine produce immunity that lives in the Boston. Okay, lives in the right places. I didn't know vaccine. Sorry, I didn't know immunity lives in lymph nodes and mucosas. I don't even know what mucosa is. Actually, I'm gonna Google it really quick. Oh, that's why the doctor's checking that stuff, right? See if it inflamed. Interesting. So it's in the lab too. Lymph nodes. Oh, okay. On the knees as well. Interesting. A lot of them here. A lot of them are on the armpits here. Usually, like when you bend, like, oh, this one is big. Cisterna Chile. Wow. There's so much to learn. So cool. Mucosa. What is mucosa? So if you have B cells and T cells from millions of antigens or billions, does this mean your what's the name, lymph nodes become bigger, or it's become bigger only when you're under active attack? What is this? Mucosa. It's I see mouse and I see stomach. Why antibodies tubes here? Like, is it even possible to comprehend all this? Because on each wall, pretty much, right? Mucosa, serosa, lymph, like people spend their entire life to get to know that. And I want to know all of this like in I don't know, 90 days or 1,000 days. We'll see. Is it like even capacity in the brain to remember all this? Includes both T cells and B cells producing antibodies. Yeah. It gets refreshed naturally when re-exposed to pathogen. Get refreshed naturally when re-exposed to pathogens. So refreshed means more B cells and T cells created, so it can keep concentration higher of antibodies. Or what that refreshed means? Costs almost nothing and has known safety profile. A T cell transfer would give particular temporary immunity as a cost of risk. Yeah, it's repeating stuff. A technique, where adaptive cell transfers is actually used. This technique called adaptive cell transfer is rare and used in medicine, but in narrow cases, CAR therapy for cancer. The patient's own T cells are extracted, engineered, and give it back to him. Oh, interesting. So the T cells reprogrammed. Donor site infusion. After a bone marrow transplant, more donor T cells are added to fight leukemia relapse with careful HLA matching. This is terrible. Must be so painful to go through this. Virus-specific T cells for transplant patients whose immune system are wiped out and who can't mount their own response. What do you mean wiped out immune system? Bottom line. Okay, then I ask you, like, what's the percentage? Always 50. Math. That's called math. I called it. I can count. A child inherits exactly half of the you inherit some fully from mother and fully from your father. So parents to child always exact 50% match. Parents different. Child to parents same things always 50. I mean, hello. This is different from siblings, where the match is probably because of each sibling and inherits one of two possible. This is interesting because one of my favorite book, what's the name? One of my favorite books is Selfish Gene. Saying that you actually more closer to your sibling than to your parent. Think about it. I love this book. 20 full match, both have 15 half match, and a chance 0 match. 20 chance full match and 25 chance 0 match. Buffing is good enough historically for bone marrow transplant doctor traditionally. Okay, I'm gonna play stupid right now. Why don't we just transport an antibodies? Let's say we take B and T cells of human A, produce antibodies in a lab. So we put them in a lab, in a dish, produce a lot of antibodies, and take those antibodies and put in a human B. I mean, besides it will cost $15 million. Oh, give me a award. You just invented a real therapy. This is, okay, AI is so hyped. I mean, I need to fine tune it to do a reality check on me, but this is essentially how monoclonal antibody therapy. You just reinvented a real therapy. This is essentially how monoclonal antibody therapy. See, it's so easy to invent shit. Yeah, just kidding. Basically, I need to retrace all the inventions that already exist, then get to the point where something not exist, and then do something that doesn't exist. The path is clear. Tap, 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 one by one. Monoclonal antibody therapy. Can they name it a little bit better? Because monoclonal and convalescent plasma works. Your intuition is correct and is used clinically, but it has important limits that explain why vaccines remain the default. Yeah, I'm not arguing that vaccine is bad. I'm just seeing convalescent plasma. Takes blood from someone who recovered from a disease, extract the antibody-rich plasma, inject No, that's not what I was saying. Inject into a sick person. It's just taking an antibody-rich plasma and put it into a sick person. I was saying to take a B and T cells into petri dish and tell them to produce a lot of antibodies, stimulate them, used for over 100 years, deployed during the flu, Ebola outbreak, and early COVID. Monoclonal antibodies, the modern industrialized version of your idea. Scientists identify a single B cells, making a great antibody against a target. Clone it, yep, and mass produce that exact antibody in bioreactor. Yes, usually using CHO cells. Some kind of, I would guess, stem cell, like neutral cell or something for base cell that can be teach to do certain stuff. Interesting. Cell banking, fuzz evaluation, clone. Really? CHO cell stands for Chinese hamster ovary cell? I'm surprised. Oh my god. A family of immortalized cell lines. What are we looking on? Oh yeah, by the way, it's explained here. Chinese hamster ovary cells, not the original human B cells. Example, rituximab for lymphoma. So yeah, so that therapy exists, like cocktail invention, pulled antibodies from thousands of donors given to people with immune deficiency. Okay, so your idea is not only valid, it's multi-billion-dollar industry. Loves this. Why it doesn't replace vaccine? Okay, I'm not asking to replace vaccine. 3 months. Antibodies are proteins, they get degraded. Yeah, it's T cells don't transfer this way. You mentioned T cells, but antibodies are only made by B cells. What does T cells do? Something different, like kill infected cells directly and coordinate the immune response. You can't battle T cells immunity as protein. And as earlier, yes, I didn't mean, like, I didn't know the function of T cells, so it's not intended. Cost and scale, narrow target production, when passive antibody transfer is the right tool. No. Okay. The storage you were reaching for. After fighting an invader, your immune system creates memory cell, a type of white blood cell called memory B cell and memory T cells. This sticks around for years, sometimes for life. They are not storing virus DNA. They are storing the shape of the antigen so they can recognize it instantly next time. It's pattern matching, not a database of genetic codes. Okay, I got confused. The database of genetic codes, it's actually On the viruses? Or bacteria? I don't know. But I remember that podcast from NBC, or whatever the station was. It was fascinating. The two layers you didn't mention. Your immune system actually has two systems working together. Innate immunity is a generic first responder. Inflammation, fever, cell that eat anything origin. Oh, inflammation, fever, cells that eat anything for origin. And cells that eat anything for origin, I'm assuming that's the T cells. No memory, no specificity, just attack anything weird. But how do they know that it's weird? I guess antigens. Adaptive immunity is a smart, slow system. It learns specific invaders, so there is two immunity: innate immunity, it's through inflammation, fevers, and adaptive immunity. Adaptive immunity, that's what's vaccination used for memory, is a smart, slow system. It learns specific invaders, make targeted antibodies, and build the memory you describe. Vaccination trains the adaptive system specifically. Scores the vaccination. Okay, what we have? 15 minutes. Question number 9. Shifting gears completely. What is the ecosystem and what happens if you remove one specific species from it? Ecosystem is a relationship between different species within a certain area or environment where it's balanced to the stable kind of degree. It's always vary, but it's more or less stable. When one species is removed, the whole ecosystem become unstable and it might die out or it might stabilize itself in a different shape. An example of this, when they brought rabbits to the Australia, they didn't have natural predators and decimated Australia. Then they brought a fox, and the fox didn't even eat rabbits because it's figured out it can eat eggs of ground nesting birds, and it decimated the birds. By removing species, like in another example, like in the forest, if you remove wolves, the deer population will go crazy, and then it might even change the shape because deer will eat too much grass. I think like Yellowstone, right? They repopulate wolf and it changes the shape of the rivers. But basically, ecosystem is a delicate balance between all participants of this system. And by removing one of participants, the whole system can become unbalanced and collapse. My best answer. The examples are doing heavy lifting. You are not just defining it, you are demonstrating understanding through cases. Relationship between species in the area, balanced but always changing, correct. Biologist calls this dynamic equilibrium. Yeah. Good job, biologist. I would call it myself if I use it, but dynamic equilibrium. Do they say it with the same sassy pronunciation?
Dynamic equilibrium.
Dynamic equilibrium
Dynamic equilibrium.
removes one species. Instability is a collapse on new equilibrium. Correct. This is called trophic cascade. Why you need a name for that, I guess? When the effects ripple through feeding levels. Australia rabbits, correct, introduces 1859. No predators devastate vegetation across the continent. Fox eating birds' eggs instead of rabbits, correct. This is textbook example. Did I read a lot of books or just listen to a lot of podcasts? Example of union unintended consequences in ecology. The introduced solution become a new problem. Yellowstone wolves, correct. This is a famous case, and this is the famous case. Wolves were reintroduced in 1995 or so recently. Fewer deer means vegetation recovers along riverbanks, which stabilize the soil, which literally change how river meandered. Wolves, deer behavior, plants, river shape. The technical terms is trophic cascade. I don't know if I'm gonna remember this.
Trophic cascade.
I think in software we can do something like this as well. Like we can introduce trophic cascade by removing or introducing one new extra microservice. And here is the most cited example. Delicate balance between all participants. Correct framing. He said it's the best answer and give me 0.95. One small addition you'd want to full picture. An ecosystem isn't just a species. It also includes the abiotics, non-living components, sunlight, water, soil, temperature, nutrition. Environment, I said environment. The wolves to river example actually illustrates this perfectly biological. Yeah, whatever. 61. Your score is climbing. The parent holds you are strong on system levels. Yes. You are strong on system level thinking, ecosystem evolution, energy, immune logic, and weak on molecular vocabulary. Nucleotides. I already forget what's nucleotides. Transcription, cell structure. I think cell structure is getting better for me. Transcription, that's what's happening from RNA to protein. From DNA to RNA. Translation, that's what's happening from. Yeah, I'm weak on. Back to the molecular side. Let's properly gap. What is a protein? What it's made of? And why do cells need them? No idea. I'm gonna guess. No, I'm not even gonna guess. I thought originally that's protein made from amino acids, but I don't think so. That's right. Or maybe it is right. And they need protein as the building blocks of cells themselves. So they basically cells are building itself or copy of itself so it can be divided from proteins. Maybe it needed to build the DNA itself or RNA. It's made of, I don't know, I don't know what's made of. Astropher's instinct more. Oh, thank you. You were right. Proteins are made from amino acids. Correct. This is the answer. A protein is a chain of amino acids folded in a specific 3D shape. There are 20 different amino acids. Yeah, I remember this. Like, I had a notepad for this. That's only 20 amino acids, and I wanted to check, and it keep 20 different amino acids, and the order they are chained together dictated by the gene determine how the protein falls and what it does. Building blocks of cells. Partially correct. Proteins are one of the main structural material, but that's only one of their job. Here's a big picture you're missing. Proteins do almost everything. Thinking of protein as a cell workforce. Cell workforce. So mitochondria, it's a cell powerhouse, and protein is a cell workforce. DNA is the blueprint library. RNA is the messenger, but proteins are the one actually doing things. A short list of job proteins do. Structure: your hair, nails, collagen in skin, muscle fiber. They're all proteins: keratin, collagen, actin, myosin. Okay. Enzymes, proteins that speed up chemical reactions. Every metabolic reaction in your body is catalyzed by protein enzyme. Without them, reactions that need to happen in milliseconds would take years. Oh. Transport. Hemoglobin, a protein, carries oxygen in your body. Immune defense, antibodies are proteins. Signaling, many hormones are proteins. Insulin, for example. Movement, muscle contraction is two protein, actin and meiosis, sliding past each other. Wow, this is so cool. I heard all these words before, like insulin, actin, meiosis, enzymes, structure, collagen, creatine, hemoglobin. But I had no idea that all that it's actually proteins. And they are workforce. So I'm thinking of comparison, like so in a world of information, if you need to transfer information from one place to another, right? How can you do it? Like right now with internet, we're doing it by light sometimes, right? Fiber optic, electricity, regular cables, 0 1 signal, 1001. This one is a little bit different because it's give you structure, scaffolding, enzyme, speed up chemical reactions. That's interesting because so what would be protein? Protein analogy in a different sphere. Immune defense, antibodies are protein, yes. Signaling, many hormones are proteins. That's interesting. Building blocks is understand. So it's a structure, enzymes. Enzymes is for catalysis. Transport, carrying oxygen, carrying basically different chemicals to different places. Immune defense, signaling, movements. There's the cells, everything that's DNA, protein matters. Okay, blocks and the machines and the sensors and the transporters and the defenders, they sell everything. That's why DNA, RNA programming matters so much. The whole point of the genome is to produce the right proteins in the right place at the right time. I love this. Your hesitation about mate of amino acids, burn this in. You have now second guessed yourself on this twice, Q4 and here. Lock it down. DNA RNA chains of nucleotides ATUGC. ATGC for DNA, AUGC for RNA, nucleotides, proteins, chain of amino acids. There's only 20 types of amino acids. There are two completely different alphabets. DNA is a recipe written in nucleotide letter. Proteins is a dish built from amino acids ingredients. Bad analogy, like really bad. Like this is confused me more. Checkpoint, 10 questions. You are roughly 60%. Here's the gap map so far. Strong evolution, ecosystem, energy, mitochondria, immune, logic, system thinking. Shaky, gene function, DNA versus RNA, information flow, but improving fast. You reconstructed the central dogma mid conversation. Weak, molecular vocabulary, nucleotide versus amino acids keep tripping you. Cell structure, organelles, protein functions. Want to keep going or take a break? I'm taking a break. That's good. We got the 10 question down in 2 days, 3 days. I'm gonna study on the weekend. And I think like few more weeks, and we're gonna be ready for experiments. Like I can't wait. Remember the learning pyramid? Like book knowledge, but it will be waste if we're not gonna apply in practice. So we gonna practice. To the next time in the class.